Good afternoon, everyone. My name is Caitlin Zapola, and together with Mackenzie Donovan, we will be presenting on energy coupling in a biological system. So just like any other activity or presentation, we always have our learning goals. By the end of today's lesson, students will be able to define energy coupling and explain why cells require it, connect ATP hydrolysis to endergonic cellular work, describe how a proton gradient stores potential energy, explain how ATP synthase converts proton flow into ATP, and predict what happens when coupling is disrupted. Before students are able to understand ATP, they first need to understand energy coupling. Energy coupling is how cells connect energy-releasing exergonic reactions with energy-requiring endergonic reactions. Instead of wasting energy released during metabolism, cells actually capture it and then temporarily store it within ATP. ATP then transfers that stored chemical energy to processes like active transport, protein synthesis, and muscle contraction. Understanding this relationship is crucial because it provides a basic foundation for everything that will be discussed next in the next slides and in the activity itself, like how ATP is actually produced. Before students complete the activity, they need to understand where ATP comes from. You're also going to notice that each step is actually color-coded. So I'm going to be using the same colors throughout the lesson to help visually connect each stage of energy coupling for those visual learners and even those ELL students. The process begins with the electron transport chain shown here in blue. This is going to pump hydrogen ions across the inner mitochondrial membrane. This then creates a protein gradient shown here in green, which stores the electrochemical potential energy. As hydrogen ions move back through the ATP synthase in orange, the enzyme then converts that stored energy into ATP. This is one of the best examples of the energy coupling because the energy is changing its form from electrochemical energy into chemical energy stored in ATP. Now we're going to take a look at what happens when the cell needs to use it. Once ATP is produced, the cell can use it to perform work. Again, notice that the same color coding continues, connecting ATP synthesis with ATP hydrolysis as one continuous process. That is very important to understand. During ATP hydrolysis, ATP loses its terminal phosphate group, producing ADP and inorganic phosphate. This then releases the chemical energy. However, the important idea here to understand is that energy is immediately coupled to endergonic cellular work. As we move into the activity, I want you to think about how ATP transfers its energy rather than simply releasing it. That is something that I want the students to do when they're undergoing this activity. Now it's time to put it into practice. This activity is going to allow students to move beyond simply memorizing the process and instead experience how energy coupling works, right? We want them moving and understanding. And sometimes when they act it out and they move along with it, it's a light bulb that goes off in their brain. So students are going to work in groups 5 or 6. Each student is going to be assigned to a singular role. We have one student take ATP, one student ADP, one student as inorganic phosphate, one as energy or cellular work. The ATP student begins by holding the energy card. When ATP hydrolysis occurs, ATP then will separate into ADP and inorganic phosphate while transferring the energy card to the cellular work student. The cellular work student will also receive one of 3 processing cards to represent either active transport, muscle contraction, or protein synthesis. This will really give the entire group the big picture. This will also demonstrate that ATP does not just release energy, it's also transferring that energy directly to an endergonic biological process. By physically acting out this sequence, students are able to visualize the connection between ATP, hydrolysis, and cellular work, which reinforces the concept of energy coupling, our main idea. After completing the activity, it's also important to make sure that students are able to connect what they experienced and what they just acted out into our phenomenon, which would be our energy coupling in the biological process. So now we have our big picture slide. This is where, as a class, we're going to come back to our main concept and bring it all together. It's the big picture. The activity focused primarily on ATP hydrolysis, but energy coupling is actually a continuous cycle. Energy from nutrients drives ATP synthesis, again shown in green to keep that showing that it's a continuous cycle. Then ATP stores that energy as chemical energy. ATP hydrolysis then transfers that stored energy to cellular work. And finally, ADP and inorganic phosphate are regenerated into ATP, allowing the cycle to repeat. The constant color coding helps students recognize that each of these individual steps is part of one continuous energy coupling cycle. At this point in the lesson, I would then guide students through the overall process before moving on to the discussion questions, just to make sure there is a basic understanding. I would also do a Padlet vote and just see where they're at with their understanding prior to moving on and just getting a basic of, yes, I'm good, no, I'm not, or maybe I'm halfway there. So here we have our discussion and our reflect questions. These I prefer to be discussed as a group, but then reflected on individually. These questions are meant to encourage students to apply the concept versus just recalling the information. Instead of asking students to define ATP hydrolysis, they're actually required to predict what would happen if energy coupling was disrupted. So, like these questions, a cell suddenly runs out of ATP, which energy requiring process would be affected first and why? ATP hydrolysis continues, but ATP synthesis stops. Predict how would this affect energy coupling and overall cellular function. A researcher develops a drug preventing ATP from transferring its phosphate group to another molecule. How would this affect energy coupling in the cell? Explain. You want those higher order questions. As students would discuss these scenarios, I would also encourage them to justify their answers, such as the why and the explain, using what they learned mainly from the activity and not so much as a definition formation. This will allow me to then assess whether students are truly understanding of how ATP synthesis, ATP hydrolysis, and cellular work are all connected. To conclude the lesson, there are always our key takeaways. There's however many ideas in this activity, it's 4 main ideas that I want the students to take away with understanding of. So energy coupling links exergonic and endergonic reactions. Protein gradients store electrochemical potential energy. ATP synthase converts proton flow into ATP, and ATP hydrolysis powers cellular work and require continuous ATP regeneration to sustain life. Together, these concepts explain how the cell efficiently captures, transfers, and utilizes energy. Now I'm going to hand it over to my partner, Mackenzie, for activity number 2.
Hello, I am Mackenzie Donovan, and for my part in this presentation, I'm going to be discussing another activity that will help with the students' understanding of energy coupling and the process of ATP hydrolysis. This activity is called Build the Gradient, and what this is meant to do is make the invisible gradient visible. Students should be able to feel that the system has potential energy. When many of the hydrogen ion students are crowded on one side, if the membrane leaks, the gradient will dissipate and ATP synthase loses its power source. So for this activity, the setup is that students are all on one side of the room and there is an intermembrane space. On the other side of the room is the mitochondrial matrix. Students are hydronium ions. Chairs or tape marks or some other distinguisher will be the membrane, and the teacher will get to pump students to one side and then open ATP synthase. I think this is really fun because students really do get engaged when they get to physically do things, and getting up and moving around creates a buffer in, more sitting and getting type of tasks. And this is the type of sort of analogy activity that would hopefully stick with them. In order to make it more effective, you could ask students, where is energy stored before the channel opens? What happens if the membrane leaks? And you can also ask them, why does ATP synthesis stop if proton flow stops? These are some things to get them thinking. To make that activity discussed a little more impactful, it should, of course, be accompanied with its lesson and examples. So, this is a good example to include. Glucose oxidation powers ATP. So, you have in the steps glucose, pyruvate, NADH, the hydronium gradient, and ATP. So the coupling chain here that they should understand and watch occur in some way with visuals is food oxidation, electron transport, proton dumping, ATP synthase, and ATP dependent cellular work. So what this would hopefully do is connect dietary energy to cellular energy. The glucose is oxidized, electrons are transferred to carriers, carriers donate electrons to the transport chain, proton pumping creates a gradient, and ATP synthase uses that gradient to make ATP. I think that this is really interesting to teach them, and while it could be complex to understand, there's something very gratifying about it, just the sophistication that requires for all of this to happen successfully. I think that students will appreciate that. And we could also create some comparisons from the activity, how there are many steps to just simply the teacher pushing the students around. The teacher has to initiate steps. The teacher has to, sort of bond with them in a way, whether they are giving them a shove or verbally telling them, making some kind of eye contact. So you can kind of make up these being steps in the similar process. An important component to the lesson on energy coupling is that it's not perfect or seamless. Coupling can be broken. It can be done by inhibitors and uncouplers. In a normal coupling, ETC pumps hydronium, the gradient will form, and ATP synthase will make ATP. But in disrupted coupling, ATP synthase inhibitors will block ATP production. The uncouplers will collapse the proton gradient. Electron transport inhibitors could stop proton pumping, or energy failure can damage those high demand tissues. Something that they should really understand, big message here, is energy coupling does make life possible, but it also creates vulnerable targets for toxins and drugs. So ATP synthase is essential, and it is conserved so that molecules can inhibit it. That inhibit it can be powerful but dangerous. Some compounds are used experimentally or therapeutically, but toxicity is a big concern because the mitochondria power the essential organs. This is one of those things that helps sort of hook the students in. Why is it important? Why do we care, right? So exercise involves ATP. The respiration will speed up to regenerate that ATP and keep you going. With nutrition, food molecules provide electrons that drive proton pumping, and with the temperature regulation of your body, homeostasis. Uncoupling releases more energy as heat instead of ATP. So this is back to a real life, daily life understanding. Anytime that cells need sustained energy, ATP, regeneration, and coupling play a role. This includes exercise, physiology, metabolism, nutrition, and mitochondrial toxicity. These are some great questions that can serve as a check for understanding and an example of critical thinking with this subject. So, a membrane becomes leaky to hydronium. What happens to ATP synthesis? You should now know this from the lesson. A cell uses ATP to pump ions against a gradient. Which reaction is endergonic? Another one. Why is oxygen indirectly connected to ATP production in mitochondria? So these can all serve as a great formative assessment. Some strong responses for it would be proton gradient collapses so that ATP synthesis decreases. Ion pumping is endergonic and ATP hydrolysis is exergonic. And oxygen accepts electrons at the end of the chain, allowing electron transport and proton pumping to continue. I would advise that to conclude a lesson like this, you have to go over the tough bits multiple times. So with that, you should probably revisit the chain of energy conversions, provide maybe another example to apply. Students should be encouraged to explain energy coupling as a sequence of transformations. ATP is a carrier and a coupling tool, not a source that can violate thermodynamics. So these are some of the takeaways that the lesson should leave them with. Cells do unfavorable work by coupling it to favorable free energy changes. ATP hydrolysis drives cellular work through phosphoryl transfer and conformational change. Electron transport stores energy in a proton gradient across a coupling membrane. ATP synthase converts proton flow into rotational motion and ATP synthesis. Disrupting coupling can rapidly cause energy failure and toxicity. This page contains the sources of our information, and on behalf of Caitlin and myself, I would like to thank you for watching and listening.